So we'll go with the next question now. Clearance. Oh, this we did many times. Do you want me to repeat? Is that all right? Better creatinine clearance.
Which question, madam?
This fourth one. Creatinine clearance.
Creatinine clearance already done now. C is equal to UV by P, that formula.
Is that all right, beta, Ritika?
Yes, ma'am.
Okay, beta. We'll go with PCR, polymerase chain reaction. Okay?
Okay, ma'am.
Please open this page number in your respective textbooks. I will also. Yeah, 534 in my book, beta. Ritika, our book 534.
Okay, ma'am.
So what is polymerase chain reaction, beta? Basically, this is the mechanism of synthesis of proteins under laboratory conditions. Basically, under normal and natural conditions in our body, we know the proteins are synthesized by a factory of mRNAs, isn't it? mRNA, tRNA, rRNA, they all will work in harmony to produce the proteins. But suppose if you undertake the similar type of process in the lab in order to synthesize proteins, then you call it as polymerase chain reaction. Did you understand the definition and all, beta? So then there are some series of steps which we need to memorize for this. 534. It's written 534, but I'm not finding it here, beta. Ritika, did you find polymerase chain reaction? Imad, can you please check?
No, ma'am.
But it's not here. So what to do now? I think polymerase chain reaction. Imad, you check in your textbook, better. Your page number.
Okay, ma'am. But my page number is addition is different.
It's fine. Just tell the chapter name. Oh, 34 only it's written here. Did you find better, Ritika, Imad?
To find.
See, polymerase chain reaction is nothing but, see, artificially there will be an instrument. Okay, inside the instrument you will give all amino acids, all enzymes which are required for the synthesis of a polypeptide chain. That means we are trying to synthesize proteins in vitro. In vitro is outside the body by creating a similar type of atmosphere which usually takes place inside our human body. So that is, for that, I think you are finding a flowchart in your books, right, Imad, Ritika?
Yes, ma'am.
You just follow the steps of flowcharts, whatever are given, and just like
Oh,
convert them into words and just write the final point, like how proteins are synthesized. Okay, Imad, you
okay,
wanted to
ma'am.
read, no? Read few points meanwhile.
Ma'am, there is no flow chart.
There is no flowchart. Okay, but I'll check in the textbook nicely and is there not any flowchart even in your? It's there, but 100%. We are not on the right page, Ritika. I'll find out and I'll share it by tomorrow. Okay? Not this chapter,
Okay, ma'am.
Ritika.
Okay,
This
ma'am.
is some other chapter, in fact. Okay. Tumor markers we have done, isn't it?
Oh,
Before that, we have
yes,
role
ma'am.
of parathormone in calcium homeostasis. Ritika, what is the source of parathormone? Parathyroid gland.
Parathyroid gland.
Okay. So you should talk about little like we have four parathyroid glands on the posterior surface of the thyroid gland. They are pea-sized, and the parathyroid glands are concerned with the release of parathormone, which what does it do? Does it increase the calcium level or decrease the calcium level in the blood? It increases. Parathormone increases the blood calcium levels. Calcitonin, which is a hormone coming from parafollicular cells of thyroid gland, will decrease the calcium in the blood. So that means these two hormones, one is parathormone and the other is calcitonin, they act in such balance that they will always maintain blood calcium levels constant in the blood. What is the normal range of calcium? 10 to 12 milligrams per deciliter. Okay, so this normal range is regulated by the hormones. Parathormone is playing a major role. Okay, suppose if blood calcium levels are falling down, it will cause bone resorption. That is dissolution of bone, and it will mobilize the calcium from the bone to the blood. Okay, so in case if already blood calcium levels are high, then it will cause the calcium to get deposited in the bone. Okay, like that. As per body's requirement, the hormone acts. Then we have tumor markers, beta. I hope you remember what are tumor markers, Ritika?
Um, the tumor markers, they determine when the before cancer, the
Yeah.
I'm not sure how to explain it now.
But
Ma'am,
you
it
gave
is
me the correct point.
those markers found in blood, urine, or tissues which,
Ah,
when elevated, shows the presence of cancers.
yeah, yeah,
For
perfect.
example, alpha fetoprotein.
Yeah. CEA, carcinoembryonic antigen. Okay, both of you
Yes,
told in
ma'am.
a correct way. So these markers will increase in blood and give the indication like person is suffering with cancer. The earliest indicators are tumor markers. If you are even suspecting after 25 years of age or 30 years of age, if there is a family history of some cancer, then we can go with tumor markers and then we can go with further investigations. Anion gap also we have done better. The small amount of anions which are generally not measured, which is actually called as anion gap. You just read once better. The reading also will be there. Functions of zinc also we have studied. Google, do you want me to repeat better? Simple only it
What,
is.
ma'am?
Anion gap, functions
No,
of
ma'am,
zinc.
anion gap already we've already came last time.
Yeah, yeah. Ritika, is that fine better?
Yes, ma'am.
Ah, zinc functions also will be very easy, better. You can just refer. Google hypothesis also we have discussed, but do you want me to revise?
Um, Ma'am, we have discussed it. I think it explains the degeneracy of genetic code,
Yeah.
universality and degeneracy of genetic code. I think we've already discussed.
Ritika, do you remember it?
Yes, ma'am. I remember
Ha.
discussing it.
Okay, nice. Very good. That means we have genetic code, no? Which are like we have three nitrogen bases, which are actually coding for one amino acid, but the last one is always different. And many genetic codes are coding for one amino acid. That means the last nitrogen base, whatever is there, that is overread. That's what is Google hypothesis. Okay. Then oncogenes, cancer-causing genes are called as oncogenes. Like we have tumor markers, there is also a table which gives us the list of oncogenes. Okay, we will see the list once.
Yes, ma'am. This
And
oncogenes.
yeah, we will see. Maple syrup urine disease is the defect of one enzyme beta, which is given in protein metabolism. Last page, there is a table which will help us to know the deficiency of which enzyme. Keep checking that. Meanwhile, I'll go to oncogenes. .687 page in my book. Yes, but see here, oncogenes. Yes. See here, you have the table. Oncoproteins are nothing but oncogenes. So you should give the list here. Just give this definition like the genes capable of causing cancer are known as oncogenes. Okay, they are actually discovered as tumor causing viruses. Okay, so they are actually very close to certain genes present in our normal cells, which are actually called as proto-oncogenes. So now about 40 viral and cellular proto-oncogenes have been identified. So here you have the list. These oncoproteins we have, proto-oncogenes we have, and what are the types of cancers it is associated with. Okay, you just read this small paragraph and give the details of this table, 37.2. Okay. So what is the defective enzyme for maple syrup urine disease, beta?
Branch chain acid keto branch chain alpha acid dehydrogenase complex.
Okay, beta. Okay, then we will stop here, beta.
Okay, ma'am.
So
Okay,
tomorrow I may take it the same time, but I'll try to take it 1 p.m. Just cooperate, beta. Okay.
Tomorrow I might have exam actually. I have exam actually
Okay. What
in the
exam you have,
morning,
beta?
10 to 1.
Okay. Then we will have at 7
Physiology.
o'clock. Okay. Right. Well, all the best. What chapter you have?
Okay. Ma'am, we have whole of respiration unit.
Okay. If you told we should have discussed respiratory system today, no, beta?
Oh, okay.
It's fine. If you have such exams, you just tell me one day before, any of you, so that it will be a
Okay.
revision for Ritika also. What is there?
Yes, ma'am.
Anyhow, we are only in the revision phase, right? We have done everything.
Yes, ma'am.
Okay, beta. So, right, well, all the best, and we will have a class at 7
Thank you.
p.m. tomorrow then.
Yes, ma'am. We'll have at 7.
Okay, beta. Take care. Bye. Good night, all of you.
Okay, ma'am. Thank you. Bye bye.